So I'm Maureen Hillenmeyer. I'm founder CEO of Hexagon Bio. I do not work on fish-free feed, or Hexagon does not, but we work in synthetic biology, and I know some of the organizers here, and we talk a lot about synthetic biology and its applications, both in medicines, as I'll talk about, and in some of the applications discussed here today. So I was invited to give a talk about Hexagon's work, and I'll try to tie it in parts in the middle where some of our lessons that we've learned I think could be applicable to making both proteins, as we've heard about this morning, but more relevantly to what we do, small molecules. So the central dogma of biology is DNA, RNA, protein, and the missing component of the central dogma is small molecules. So proteins usually make various small molecules and metabolites throughout cells. So some of the medicines that we make today, or some of the lessons from making those types of medicines and small molecules, I think could help some of the types of projects and companies in this area. Okay, so I'll give a really quick aim for 10 minutes with 5 minutes of questions, so just run through really high level what Hexagon Bio does. So I worked for many years at Stanford before Hexagon on the science that became the platform of Hexagon. And my co-founders and I spun it out about 2 years ago from Stanford to Hexagon. Just quick background, we're based in Menlo Park, California. We have 22 employees, about half computational and half experimental. And the experimental folks are mostly biologists and chemists doing some of this synthetic biology platform work. So we use data science and synthetic biology to discover and develop new medicines. So at Hexagon, we work on medicines called natural products. So these are medicines from nature. Famous examples include penicillin, the first antibiotic. This was discovered by accident. People probably learned about it in high school biology. Alexander Fleming, this guy in the early 1900s, was growing bacteria on a petri dish. A fungus flew in, started killing the bacteria. He and others eventually isolated the active component, and that was penicillin. And that's really how almost all antibiotics were discovered subsequently, was trying to repeat that happy accident, so letting things grow next to bacteria, see what would kill the bacteria, then isolate the active component. Many other drugs were discovered in this manner as well, especially as infectious disease started to come under control in the 1950s and 60s. People turned their attentions to other diseases. So lovastatin, the $100 billion cholesterol drugs over their lifetimes, also discovered in a fungus. And the reason is that these things didn't evolve to help people or to help in the clinic. They evolved really selfishly. These bugs are trying to kill each other off in the soils and the rainforests and the oceans. So these are kind of competitive, we call it chemical warfare molecules, where these bugs and plants are trying to kill each other. So lovastatin is a case, probably evolved originally as an antifungal, inhibiting cholesterol so potently is actually toxic to fungi. And it just so happens for people, lowering cholesterol is useful for cardiovascular benefits. So we've just repurposed these drugs over and over from nature throughout history over the last several decades. So about 3/4 of all FDA-approved antibacterial drugs are from natural products, so microbes and plants, and 49% of anti-cancer drugs are natural products. So next time you or someone you know is prescribed a medicine, look it up on Wikipedia. There's a good chance it will have been found from a fungus or bacteria or plant. Okay, natural product drug discovery is challenging, however. I hear big pharma companies like the Pfizers of 40 years ago used to give employees bonuses if they would bring back samples from some crazy place in the world because it might have a new species that would lead to a new drug. Unfortunately, most species will not grow in a laboratory environment. So this is a major problem with natural product drug discovery historically. So Hexagon brings a new approach to this, and it's really enabled by DNA sequencing. It's something I haven't heard a lot about this morning, but I'm sure it's affecting at least the synthetic biology-related projects here. DNA sequencing, the changes there are a big deal in the whole field of biology. I'll have a graph in a minute showing the exponential increases in sequences as the costs have dropped exponentially. But in our case, natural products are encoded by genes, so DNA sequencing is very important because we read DNA. I'll try to avoid the pointer, actually, since we have 3 screens. So we read DNA from all of these new bugs that are getting sequenced around the globe. And we identify genes. And in our case, like I mentioned, we're interested in small molecules, not just a protein itself, so we have to go that extra step. So genes encode proteins, which make these small molecules. So the one shown here is lovastatin. And as I mentioned, DNA sequencing is getting easier and easier. So people around the world, right now mostly academics, people at universities, are sequencing the Earth. Illumina is the main technology, but there's others like PacBio and Oxford Nanopore. People are going out into the field with some of these technologies and just sequencing on the fly. So the number of DNA sequences is really going up exponentially, and the databases are growing rapidly. So right now, this year, at Hexagon, we focus on fungi, so I'll just give some numbers on fungal genomes. There's about 3,000 fungal genomes that have been sequenced as of 2019. There's an estimated 5 million species on the Earth, and the major species of interest for drug discovery are fungi, bacteria, and plants. So just in the fungal kingdom alone, 5 million species. So there's a lot of undiscovered potential out there, both in medicines and in enzymes and proteins that people here would probably be interested in. But unfortunately, as I mentioned at the beginning, 90% of these bugs will not grow in the laboratory. So this is the part that I think is useful beyond just Hexagon's work in making medicines. We spent a long time, my colleagues and I at Stanford, getting genes to work well in a yeast host. So this is just baker's yeast, Saccharomyces cerevisiae, beer and bread yeast, the exact same species. This is a favored organism in both biology as a model organism because it's very well studied, and even as a workhorse industrially. So again, we start by reading DNA from these native bugs. We just heard mention of Aspergillus a minute ago. They make a lot of these useful medicines. So we read their DNA. That becomes genes in a database. And then importantly, I'd say a second enabling technology for Hexagon and again, probably other synthetic biology companies, in addition to DNA sequencing, is DNA synthesis. So we no longer have to clone laboriously, working with these native hosts and get their DNA. We can simply print DNA. So there's a lot of DNA synthesis companies around the world and even a lot in the Bay Area that we at Hexagon work closely with. So we basically read DNA from the cloud, and then we print DNA at a third-party provider, and we insert that DNA into our baker's yeast cells, and now we're forcing yeast to make these chemicals as opposed to those native hosts. And so this is how we get around the problem that most of these species will not grow in the laboratory. Yeast grows really well, and all we need is the DNA. We do have to edit the genes slightly, and this is, I'll just go back a slide. The reference at the bottom here is my colleagues in my paper that we published after several years of work at Stanford. So the details are described there. I don't have time to go into it now, but basically we came up with a new strain of Saccharomyces cerevisiae that starts expressing these genes, these non-native genes, after about 24 hours of growth. So we found that if we forced these cells to express these non-native genes right away, they were very sick. They would figure out clever ways, evolution would figure out clever ways to delete the genes, get rid of them. But if we let the cells grow up slowly over 24 hours and then force them to turn on these genes, suddenly we would get a lot of these compounds out. And we published in this work, we did find a new molecule, or several new molecules. So we attempted about 40 pathways from 40 weird fungi from around the world, again, rainforests and oceans and all over. And 20 of the 40 actually made a molecule. So that's glass half full or half empty. We were thrilled by that. That was better than anyone had ever seen before. And we felt we had unlocked at least half of this mountain or underneath the iceberg of molecules. So this is just to show a number of genome sequences are rising rapidly. This is kind of the inverse of the graph everybody shows of DNA sequencing costs going down exponentially. It's a very exciting time in biology. So at Hexagon Bio, this is our platform, which we developed at Stanford and licensed out. So we start with fungal genome sequence. We mine what we call BGCs, or biosynthetic gene clusters. That's the computational component that I didn't have time to get into today. BGC selection, we select which genes we have predicted are going to be making useful medicines. BGC refactoring, that's what I talked about, so putting on different promoters in this special yeast strain that waits basically 24 hours to force these genes to come on. And that's cluster expression, which makes the proteins and subsequently the small molecules. And then we look at presence of the molecule. So we are focused on medicines. We have a pipeline of a few different medicines we're pursuing. We're preclinical stage, hoping to get into the clinic in a couple of years. All right, I'll stop there for any questions. Thank you.
Thank you, Maureen. We got time for a couple questions?
Thanks. Alan LeBlanc with Callista. So it seems to me like the secret sauce is identifying which gene sequence codes for the novel molecule. Yep. How do you find the right segment when you don't know what you're looking for as a product?
Yeah, so that's the computational half, which I didn't have time to go into, so thank you for allowing me to go into it. So we, at Hexagon, we don't know the structure of the molecule that we're making. All we have are those DNA sequences, so we don't have that, so we don't have that structure at the end. So we look at the DNA and look for signatures that tell us what this microbe might be making. So I like to use an example of the statins, the cholesterol drugs. It's made by a fungus, and we figured out while we were at Stanford and looking through these genes that we could have predicted that that fungus was making a cholesterol inhibitor. And the way it works is actually fairly simple. So there's always this mystery of how these bugs protect themselves. People thought, these are toxins, these bugs are trying to kill each other. Maybe they're just pumping these molecules out. It turns out there's a second way, which is sometimes they mutate the target protein of the small molecule. So statins are known to inhibit this cholesterol protein called HMGR. And people sequenced the genome of the fungus, and we noted that the HMGR protein, the cholesterol protein in that fungus, was mutated in a way that would have told us, if the statins had already been discovered, that this fungus was making a cholesterol inhibitor. So we simply look for mutations in certain target proteins that tell us that fungus is making an inhibitor of that protein. There's a lot of proteins involved in cancer. So at Hexagon, we're initially focused on oncology.
Thank you.
All right. Thank you. Thank you, Maureen.